Hi there, in this tutorial I'll show you how to look through public experiments and how to interpret what you see. Let's start by browsing through the table of all the public experiments available in the database. And we can sort this table by date, owner, or name. You can search it for certain terms of interest. For example, I look here for all the RNA-seq experiments in the database. And I can open any one we like. For example, I'll pick here this experiment based on data by Caput et al. 2011, which combines some RNA-seq and ChIP-seq data in one source. The data has been downloaded from the Sequence Week archive, and the original source is properly linked in this experiment. There's also an abstract for the original research publication and a link out to the original publication as well. Moreover, there are link outs to the gene expression omnibus, the corresponding entries in there. It says here the experiment has been analyzed by myself, 27th of March 2012. There's a link to the history of the analysis available, and we can export the entire information in a PDF format. I could now browse the genomic data from this experiment in the genome browser, but let's not do this. There's a separate tutorial available that shows you how to work the genome browser. Instead, let's stay on this page and see what else is there. So here's a listing of all the input data that went into the process. This is what we obtained from the Sequence Read archive. We can look at any of the datasets or download them. Next, there's a section with some annotation about the input data. You can see that the 6 datasets come from 6 different experimental samples, 3 RNA-seq experiments and 4 ChIP-seq experiments. The ChIP-seq experiments were for targeting a transcription factor called FOXP1 in embryonic stem cells of the H9 cell line, and the RNA-seq datasets were from a control cell line and from 2 knockdown experiments. Now, in the next section, we can see the main experimental results from this analysis, as chosen by the person that ran the analysis. First, we see some genome browser snapshots highlighting regions of particular interest, and we can click any of them and browse in detail what's going on. Below this, there are the main output datasets. These are, on the one hand, summary reports at various stages of the analysis process, like, for example, these sequence summary statistics that tell us something about the input data. So we see the sequence composition, the length of the reads in the individual input libraries. This all looks pretty okay here, quite uniform. There are some quality scores along the length of the reads, again that looks quite good. It's a summary of the alignment process and how well that works. You can see how many reads there were aligned in total. How they were spread out over the individual chromosomes of the genome. And how ambiguous the alignment was. For the 3 RNA-seq datasets, we are allowing up to 10 possible alignments, and you see about 60% were aligned uniquely, the rest then with a certain amount of ambiguity. ChIP-seq datasets, only unique alignments were accepted, but about 80% of the reads could be aligned. This is a summary of the ChIP-seq binding data, the enriched events that were found. Most of them were found somewhere in the narrow promoter regions of genes, and the tutorial describes exactly what we mean by a narrow promoter region. You can see here in the documentation of the corresponding module, that means everything within a radius of 500 base pairs around the transcription start site. We also see another plot detailing in more resolution, in a higher resolution, where these binding events occurred. There's a similar summary for expression data available, but let's look at one of the tabular output datasets. So we have here this blue dataset, that's gene-centric data, and in this example it combines the FOXP1 ChIP-seq data with the RNA-seq data from the same experiment. So we see there's a whole lot of columns with expression data, but also with fold changes between the different conditions and p-values calculated using DE-seq. We could browse this dataset in detail, and there is another tutorial available that shows you exactly how to do that. Now, what else is on this page? You can export the data, either the tabular data individually or all the genomic datasets in batch. Now the last thing that I'd like to show you is at the very bottom of the page. There's a simplified summary of the entire analysis workflow that was used to analyze the data in this experiment. And we can open this in a more detailed view by clicking this Workflow Designer button. So you see this complete overview of the entire analysis process. On the left there are the input datasets that were uploaded to the experiment. And then there are lots of boxes and arrows. Each of the boxes represents one step in the analysis process. So we can track down for every individual dataset exactly what happened to it. For the individual analysis processes, you can even look up the parameters that were set. So it's really completely transparent and reproducible. And all the intermediate datasets are available as well, so you can open them. There's also a history of all the actual runs of the analysis modules. We can open log files for each individual process and see exactly the command and output that came out at the time when the process was run. There's also another link to this experiment history available direct from the main page, so you don't need to go to the Workflow Designer first. Okay, this is all I wanted to show you in this tutorial. I hope you found this informative, and I'll see you next time. Bye bye.